Welcome to the video series on biotechnology and molecular biology. My name is Pranav, and today we're going to be talking about transcriptional regulation as well as post-transcriptional regulation. So in the last video, I talked about DNA, RNA, protein, the central dogma, the structure of all these molecules, and a lot of the basic fundamentals of molecular biology. In this video, I'm going to expand upon the concepts relating to transcription and the regulation of transcription. And you can see that on the screen over there. So first off, I'll talk about transcriptional regulation in bacteria. Bacteria and prokaryotes have operons, which are these bundles of genes that are located spatially together on the strand of DNA so that they can all be regulated in the same way. Oftentimes, these different genes relate to a similar function. So if all these genes are needed to do one thing, then it would make sense to have them all turned on at the same time or all turned off at the same time. You can see on the slide a diagram of the lac operon. This is a very well-studied operon in, I think, E. coli and probably many other bacteria. In the middle there, you have a promoter. That's where RNA polymerase binds. RNA polymerase, again, is the enzyme that does transcription, right? It tracks along the DNA and creates a copy of RNA from that. On the left-hand side, you have a cap site. CAP stands for catabolite activator protein, and catabolite activator protein is what binds to that cap site. It's an activator, which means that it promotes the binding of RNA polymerase to the promoter. So when CAP attaches to the DNA upstream of the promoter, it helps RNA polymerase latch onto the promoter. And downstream of the promoter, right ahead of the promoter, you have an operator, and that's where the repressor binds. So if the repressor is bound to the operator, then RNA polymerase can't move forward. Like it literally stops it from moving forward, just like spatially. Then ahead of those, downstream of those, you have LAC-Z, LAC-Y, LAC-A, and those are the genes that are transcribed from this DNA. So the activator on the left and the repressor on the right together allow the bacteria to regulate the transcription of LAC-Z, LAC-Y, and LAC-A, depending on the circumstances. So in eukaryotes, transcriptional regulation is way more complicated because our DNA, so we're eukaryotes, our cells are eukaryotic cells. Our DNA is not organized in these neat little operons. Our genes that are closely related, that have similar functions, are spread out throughout the genome. So this requires complicated mechanisms like you can see on the right-hand side where the DNA bends and several different activators and repressors and mediator proteins all come together to influence the probability of RNA polymerase binding to the promoter and transcribing a certain gene. There's chromatin remodeling. If you remember from the last video, I talked about chromatin, which is the compaction of the DNA. DNA has these histone proteins on it, and because of that, it can fold up and get really tight and even form chromosomes. So what chromatin remodeling does is it can relax or tighten the packing of the chromatin, and as a result, it can change how easily accessible the DNA is to RNA polymerase. Transcription factors regulate how probable it is for RNA polymerase to bind to the promoter, and epigenetics relates to chromatin modeling, where you can have these chemical tags attached to the outside of the DNA. So the DNA bases aren't actually changed, but certain aspects of chemicals attached to the DNA bases are changed. An example of this is methylation, which depending on where it's at, it can increase or decrease the rate of transcription of a gene. What I just talked about before was transcriptional regulation. That regulated how likely it is for a certain gene, a certain piece of DNA to be transcribed into RNA. But even after a gene is transcribed into RNA, there's more regulation. In eukaryotes, we have a thing called alternative splicing. This involves introns and exons, so introns and exons exons, and each gene has multiple introns and exons, and the exons are put together and expressed, while the introns are spliced out and just kind of degraded. So you can see in the picture on the right, you have a red block, a yellow block, a green block, a blue block. On the right-hand side, we would call the yellow block an intron because it's just thrown away. And we'd call the red, green, and blue blocks exons because those are put together and actually expressed, used to make protein. Whereas on the left-hand side, the same gene is alternatively spliced to have the red, yellow, and blue blocks or sets of DNA as exons, while the green block is an intron, and that's spliced out, that's degraded. So from the same gene, from the same uh, piece of DNA, you can actually have multiple different protein products being created. And this allows us to accomplish a much wider variety of functions and structures. And it's crazy. It's really cool. Now, mostly in eukaryotes, we have 5' capping. This involves a tagging of a modified guanosine base, DNA base, added to the 5' end of the RNA, along with 3 phosphate groups. You can see that on the right-hand side. And this helps with nuclear export stability against like degradation because RNA is quickly degraded in cells. So it helps with stability and it helps with translation promotion too, with binding of the ribosome to the RNA to translate into protein. Polyadenylation is similar, except it occurs on the other side, on the 3' end. Um, and this helps with the same things, nuclear export stability, translation promotion. Polyadenylation is exactly what it sounds like, poly, which is many, and adenyl, which is like the adenine DNA base. So you get many A's, it's just like and you can see that on the right over there. Now, another way that cells regulate how much of a RNA product is created is through miRNA. So the stuff about transcriptional regulation that I talked about before was before or while transcription was going on. miRNA, on the other hand, takes place after transcription happens. So it silences transcription by binding to complementary mRNA. If you remember from the previous video, DNA and RNA have 4 bases each, and these bases each have a partner that they base pair to, right? So in RNA, adenine base pairs to uracil, and guanine base pairs to cytosine. So the cell creates an miRNA, and if that miRNA is fully complementary to an mRNA that the cell has made, then the mRNA uses some complicated proteins like the Argonaute protein to just chop up that mRNA, and that stops that mRNA from being translated into protein. So then it's useless, it's been silenced, and that gene will not be used while that's happening. If the mRNA is only partially complementary to the mRNA, so not every single base pairs with another base on the mRNA, just some of them or most of them do, then there's a slightly more complicated mechanism by which it's not chopped up, but it's sequestered. It's put away and silenced in a more indirect way. So that's all for today. Next time we'll be talking about plasmids and plasmid engineering, which gets more heavily into the genetic engineering aspect of this. So all of this stuff that we've talked about in the last 2 videos has been the fundamentals of molecular biology. And going forward, we're going to be stepping more into biotechnology. So as always, happy learning.